Okay, so let's start today's lecture. So today is the 8th lecture, and today we are going to talk about metagenomics. So we have kind of gone past the analysis of single genome. So we're going to increase the complexity a little bit and consider the situation where we have more than one organism or more than one source of genomes in the data. So today's agenda, we're going to talk about two things. The study of microbiome and the metagenomics techniques, and the new terminology and the new algorithms that we need in order to analyze kind of these more complex data. Okay. So it's not that different from when we talked about genomics and DNA sequencing from the past lectures. So metagenomics is basically the analysis of a collection of organisms. So you're going to assume that in your sample, there is more than one genome. For example, from tumor, usually we think of tumor sample as just human. But different cells in the tumor have different genotype. And in today, many people start to realize that the tumor microenvironment, like the environment surrounding the tumors, can also synergize with certain bacteria. So we can identify more than just human DNA in a tumor. And if you study kind of gut microbiome, either from human or animal, we study the stool sample. If you are more toward like a plant or environment study, then we can study the soil, the water, and maybe the factory waste, so that we understand what impact animals and us are making on the environment. So there are two primary ways to characterize a metagenomic sample. You can focus on specific genes. So amplicon sequencing means that you are amplifying a certain gene, like ribosomal RNA, and study only that gene. So it's going to be not so complicated. But sometimes you want to study every gene in the sample. So that becomes a full-blown shotgun metagenomics analysis, where you study all DNA fragment or RNA fragment in the sample. So that can be much more complicated than a single genome situation. So before we kind of dive into all the details, so let's kind of take an overview of what difficulty can arise when we have a metagenomic sample. So first, the obvious one, all the reads that we are getting now can come from different genomes. How do we distinguish them? Whether this is a human read or this is a viral or bacterial reads. So in the clinical situation, usually it's just human or host versus the pathogens. But if you are studying environmental sample, now you start to have a mix of animal DNA in the environment and also the microorganisms. So you can have hundreds of sources of DNA in a single sample. In this context, sequence alignment now is not viable, because you cannot hope to produce a complete set of reference genome. If you want to do alignment, we have to have the reference genome. But if the sample consists of hundreds of microorganisms and animals, most of them we don't even have the reference genome. So now assembly will become the primary technique. We need to be able to assemble the reads in the data in order to reconstruct the full gene sequence or even the full bacterial or viral DNA. So you can see that for metagenomics, using short read is going to be very challenging. With long read, it's going to be much easier to reconstruct the whole pathogen genomes. And the problem can be even more difficult if you imagine that in the case of bacteria or virus, in the sample, there might be more than one strain. So it's not only the difficulty of having different species. Sometimes you can have the same species with slightly different variants, slightly different strains in the same sample. So it's even more difficult to distinguish how many genotypes you have in your data. So let's look at some example of microbiome where we want to perform kind of metagenomic analysis. The popular one, the most popular one, is probably the gut microbiome. And we start to appreciate more and more that in our body, we have more bacterial cells than our cells actually, and they are synergized with our body. They can digest food, and then they can produce nutrients and metabolites that our body can absorb. So it's a win-win situation. And as a community, bacteria that are in our gut, in our body, can also protect against pathogens, because pathogens and bacteria that we have can be incompatible. Like they might compete for the same nutrient, and new pathogens have less population than the normal bacteria that we have, so they can keep the new species or these pathogens to the outside of our gut. So now there people are using more and more of gut microbiome as an indicator of the health, either for the human or for the animal. And for human in particular, there is the Human Microbiome project where people want to understand the variety of bacteria that lives on our body, all over the organs, both internal organ and on the skin, in the respiratory tract, everywhere in the body. And the bacteria are different. Different bacteria live in different parts of our body. And people who joined the Human Microbiome project made their data available on the project website. So nowadays, I think there will be even more than this, even more than 30,000 samples. So we can characterize the variety and the distribution of microbiome, at least in human, to a very deep scale. For people who study plants, we know that plants have to exchange nutrients like nitrogen with bacteria in the soil. So people now start to appreciate that if you want to grow certain plant in certain region, it's not just about the nutrients. Sometimes it's about which microbiome, which bacteria has to be available in the soil. So you can condition the soil by putting in specific bacteria that can help certain crops grow better or produce more output, more fruits. In terms of medicine, so people start to appreciate that in the tumor environment, because cancer develop new mutation and they grow much faster, they require more nutrient, more metabolite. And sometimes certain bacteria can colonize this environment by producing the cancer with the required molecules. So that helps the cancer grow even further. And the presence of bacteria surrounding the tumor tissue can sometimes interfere with our body's immune response. So it helps protect the cancer from our immune cells. So nowadays people are starting to do metabolomics to study the metabolite surrounding the cancer tissues and understand and also perform the microbiome metagenomic study in order to understand which bacteria can serve as a biomarker for a certain type of cancer. And we can modify these, like if we kill certain bacteria, if we can target certain bacteria, it can disrupt the ecosystem where the bacteria and the cancer grow together. So this is another venue for treating cancer by changing the bacteria that help the cancer survive. In another study from 5 years ago, which was pretty exciting at the time, so people try to understand if there is any microbiome factors that distinguish between people who are good at athletics, people who can run faster, people who can jump higher. And they study the gut microbiome differences between athletes, so people who do sports, and regular people. And what they found is that people who are good at sports have certain bacteria that can utilize lactate. So lactate is what our muscle produce when we use energy. When we utilize energy, utilize glucose, we produce lactate as a product. And when bacteria in our gut, if they can utilize lactate, these bacteria produce beneficial short-chain fatty acid, which our body can absorb and make our muscle perform better. So they identify this bacteria, and then they want to be sure whether these bacteria are responsible for the capability of athletes. So they study in mice, they transplant this bacteria into the mouse gut. And they identify that mouse without the bacteria perform worse than mouse with this bacteria. So they can let the mouse run through their toys like in their cage, and they see how long does it take for the mouse to get tired. If the mouse can run for longer period, that means the mouse is getting better as an athlete. And they found that by implanting this specific bacteria, they can change the running capability or the strength of the mouse very clearly. So it was an exciting study at the time because you can modify a person's ability by just changing the gut microbiome. In the clinical context, we talked about nanopore sequencing being a rapid solution that you can identify pathogens in blood or urine within hours. So nowadays, people are starting in many hospitals in the world. They're trying to study if nanopore sequencing can become a standard bedside testing. So you can collect data, you can collect blood or urine sample from the patient, run it through the pipeline. Within 3 or 4 hours, you can process the sample, run it through the nanopore. Within 5 to 10 hours, you can have the variant annotation. You can have the gene detected. And within half a day, you can inform the clinician which antibiotics should be used to be appropriated with the pathogen species and the antimicrobial resistant genes that are found in the sample. If we use like a traditional culture-based method, like we have to culture the bacteria first, let it grow, and then treat it with drugs to see if it's resistant or susceptible. That usually takes days. So people are exploring if we can reduce the amount of time before we can select the antibiotics from several days down to half a day with this pipeline. For people who do more like an environment or kind of wildlife study, we are starting to try to understand the dynamics in the forest, like without having to be there, without having to actually getting the sample from the animals. We can collect the stool sample from the animal and look at the gut microbiome inside. Because different animals eat different food, and they might have different gut microbiome that help digest those specific food. So by studying the gut microbiome in the stool sample in the forest, like if we collect them, we can understand the diversity of animals that are roaming in the forest. So we can essentially monitor the progress of the nature without having to be as invasive as catching the animal and collecting the blood sample. So this can help with the conservation and with the monitoring of the condition for the ecosystem. So nowadays, especially after the COVID-19 pandemic, people are starting to promote the One Health concept, where the defense against new disease doesn't involve only human, but it involves animal. Because we interact with animal, we eat animal, and also the environment. Because new virus can emerge in the wild, in bats, in some cases, like after a certain time, it can transmit to human. So the concept of One Health is that we need to study the metagenomics to understand the change in pathogens in wildlife, in livestock, in agriculture before they transmit to human. Then that's too late. And when we get a pandemic, we need to keep monitoring the changing evolution of the pathogens so that we know what could be the possible mutation that can occur in the future. Okay. So just to summarize, the benefits of being able to do metagenomics, we can use it for health application, like try to identify microbiome markers for certain diseases like cancer, or to understand if a patient will respond well to drugs or treatment, because bacteria can process or digest the drugs. For ecology, we want to know how our action affect the environment. So we can monitor the bacteria, the changing in bacteria species inside factory or hospital waste. So we know whether the use of certain chemical in the factory or the use of certain antibiotics in the hospital, does it affect the bacteria that we are releasing into the environment? And also the effect of global warming. Do we get people are saying that we get more pathogens, new pathogens, because of the changing climate. As the earth gets warmer, new bacteria that can survive better in warmer temperature start to arise. So So we see a change in pathogens from before. And in terms of biotechnology, many new tools, many enzymes that we have nowadays comes from microbiome metagenomic study of bacteria in extreme environment, like in the volcano, in the hot spring. So we get enzymes that are resistant to heat, for example. So one thing that we should appreciate about the microbiome and the analysis of microorganisms with metagenomics is the idea that it is very different from large animals like human. Right? When we think about the distribution of human or the evolution of human, we are limited by our geographical location. People in Thailand will have the different genetic background than people in the US or in Australia. But for bacteria, it's different. Many people have the theory from long ago that bacteria can go anywhere. The wind can blow the bacteria. Maybe the bacteria is caught in the water droplet and it can travel with the rain and go far away in the earth. So people have this theory that every bacteria can be everywhere. There is no boundary. But the difference between bacteria and us is that we and other large animals, we can withstand changing environment. We can withstand changing temperature. You can survive in sub-zero degree and also 35 degree, but bacteria probably cannot. So large animals like us can withstand changing environment, but we cannot travel very far. I mean, nowadays we can fly, so we can travel very far, but other animals are usually fixed or constrained to a certain geographical area. And that limits their evolution. But for microbes, they can travel everywhere. But if the temperature becomes too hot, then some of them die. If the humidity changes, some of them die. So the bacteria can always travel. But whether they survive or not depends on whether they can withstand the environment. So the dynamics, the study of evolution, the models of evolution for bacteria and for large animals will be very different. And we will talk about that a little bit. Okay. Another thing is the microbiome is extremely dynamic. So there is many study about gut microbiome in human, and you might expect that different human, once we became adult, our gut microbiome might be stable unless we got sick, unless we got food poisoning, or we ingest some toxin. Our gut microbiome should be stable. But in reality, from birth until adulthood, our gut microbiome change all the time. When we are born, we have the microbiome that exists in the womb. But once we are outside, we now are exposed to food, to antibiotics, and to the breast milk. And people are starting to realize that microbiome can be transferred from everywhere into our body. And in some study that I analyzed, the gut microbiome in an adult human can change over the course of a few months. Like next 2 months, depending on what you eat, your gut microbiome might be totally different from what you are having today. So that they are extremely dynamic. So the study have to kind of involve a lot of controls and a lot of time points. And also people found that saying gut microbiome is a very simplistic description. Inside our body, different regions in the gut and the intestine, we actually have different microbiome. So even though we say that we each have a gut microbiome, it differs depends on where you look inside your digestive tract. So there are so many variables, so many parameters about this. Okay. So first, let's talk about some change in concepts in terms of the evolution of bacteria. So for large animal, for human, we usually have a very clear concept of taxonomy, like genus, family, species. But for microorganisms, many of them are so similar that you cannot clearly distinguish them into separate species. Sometimes you need to consider them as a group. So operational taxonomic unit is a technical term that describes a group of genomes where we cannot distinguish them. We have to group them as a unit. So we just say that this is our operational unit of the taxonomy. We don't have a better way of describing them. So the difference comparing to what we learned from the evolutionary lecture, like to microbiome, is that usually we expect the genome to be clearly different between different species, between different organisms. But for bacteria, sometimes they are so similar that you cannot draw crates or draw boxes like this. They are so all similar to each other. So how do we draw the cutoff? How do we group them into units, into related organisms, so that we can analyze maybe their function, or maybe we can identify biomarkers. So let me give you an example where similarity in the genetics alone is not enough to classify a bacteria. So some of you might be familiar with the genus Shigella. So this is a genus that evolved from E. coli. So E. coli is usually harmless. It's the bacteria that we have in our body. But the Shigella genus, this is pathogenic. These are pathogens. So they diverged from E. coli a while back, but they are still very similar, 80 to 90% similar to E. coli. And if you do a typical phylogenetic reconstruction, you're not gonna be able to separate Shigella from all the E. coli around them. So this raised many people who study phylogenetics or evolution of bacteria to think that maybe genome alone, like genotypes alone, is not enough to study bacteria. We need to think about their phenotype, or their function, or their ability, or their genes as well. Okay. So people start to think, when we try to group bacteria, instead of grouping them based on genotypes alone, like the regular phylogenetics that we learn about, we need to think about their function. Like, are they resistant to the same drugs? Do they exist in the same environment? And so we need to take the phenotype, the function of the bacteria into account as well. So that can make the phylogenetic analysis even more complex. But it will allow us to understand bacteria that are actually related when the genotype alone cannot tell us that information. So several people try to come up with a guideline, like how similar are the bacteria? Should we call them from the same genus? How similar must they be in order to call that they are from the same family? So in the case of a single gene, like ribosomal RNA, like 16S ribosomal RNA, we have a rough guideline. So if you see two ribosomal RNA that are about 95% similar, then you can probably call that these genomes are from the same genus. And further, then you can call that they are from the same family, from the same phylum. But these are all empirical. So people study known ribosomal RNA sequence and try to come up with this cutoff. So to study the microbiome, like we have two choices. One is the amplicon sequencing, where we pick a gene and we analyze. And that's simple. So we're going to look at that pipeline first. So we're going to start by looking at how we analyze ribosomal RNA data from the microbiome sample. So just to recap, we mentioned this briefly in the first few lectures. So in prokaryotes, their ribosomal RNA, the 16S subunit, have a very interesting structure. So some part of the ribosomes are very conserved. These blue regions are highly conserved. But the yellow regions, these are variable. So different prokaryotes will have different DNA RNA in the yellow region, but they pretty much have the same DNA RNA in the blue region. So for us, we can design primer that can amplify certain hypervariable region, like certain yellow region, by designing the primer to be specific to the flanking conserved region. So we have a choice. Right? We can amplify the V1, V2 hypervariable region We can identify V3, V4 region, V5, V6, V7, V8, V9. So the first question that people ask is, do they make a difference? Can we just pick one, or some of them give us better evolutionary information or taxonomic information than the other? We should always ask this kind of question. So long ago, people did some analysis and see if we use sequence that we get from the V1, V2 region, or from V1 all the way to V3, or the whole sequence like V1 to V9, or just V3 to V5 or V4. These are some possible choices of the sequencing. And of course, if you have a longer read, you're going to see more, you're going to be able to distinguish more organisms. The y-axis here is the number of OTU. So the more OTU you see, that means you have a higher resolution. You can distinguish two organisms apart. So if you have the whole region, like V1 to V9, you can see the highest number of OTU. But if you have just V1 and V2, or V1 up to V3, like you are losing more than half of the sequence, you're not losing a lot. You're losing maybe less than 10%, less than 5% of the power to distinguish between organisms. So nowadays, when we perform 16S RNA sequencing, if you do long read, it's going to give you the whole sequence. But even if you do short read with Illumina, and you get only 2 of the 9 hypervariable regions, you're not losing a lot. So people nowadays are not sequencing the whole thing. They are sequencing maybe 1 or 2 hypervariable regions, and that's good. But from here, like you can see that the choice of which hypervariable region can be important. If you study V4, like, or V3 to V5, seems like you are not going to be able to distinguish organisms as good as if you study V1, V2. So this information can help you decide which area to sequence in your study. Okay. And because if we decide to study just one gene, especially for ribosomal RNA, nowadays we have a lot of knowledge about ribosomal RNA. So if you go to the BLAST website, where we did some search before, there are actually specific options for you to select just ribosomal RNA. So when you have a new metagenomic sequencing for ribosomal RNA, you don't even have to do assembly. You can do alignment. Because for these specific genes, we know so much about them. And these are some other databases that you can go and download all the known RNA sequence from the bacteria or virus or fungi, SIWA, green genes, and RDP. And this is just to give you an idea of the number of ribosomal RNA in this database. Nowadays, we have almost 10 million ribosomal RNA sequence in the database. So sequence alignment becomes practical for at least for this gene. Okay. So not many of you probably will study fungi, but just to give you the information, in fungi and algae, we are also using rRNA, but we are not sequencing the rRNA itself. So in bacteria, we are sequencing the part of the 16S ribosomal RNA. But for fungi and algae, we are actually sequencing the intergene region between the 18S subunit, the 5 and 8S subunit, and the 28S subunit. So people are actually sequencing this internal transcript spacer, so the spacer between ribosomal genes. So these are regions where different fungi and algae will have different DNA sequence. So, but the concept is the same. We can do short read or long read sequencing of these two regions, and then we can either do sequence alignment or classify the taxonomy. So I mentioned about the fact that we can do sequence alignment to map our 16S RNA sequencing data to the taxonomy. But I'm going to also introduce another technique where we can use machine learning, like a data-driven approach, to classify sequencing read that we got from 16S RNA into taxonomy. So the concept is going to use what we call a DNA Kmer. So for those of you probably have seen Kmer before. So Kmer, similar to in chemistry, right? If we have a monomer, then it's a single unit. If we have a dimer, then it's a 2 unit. So Kmer is a K unit of some monomers. In this case, the monomers are the nucleotide. And Kmers mean that we are considering a nucleotide of length K. They are K nucleotides. So this is similar to when we learn about BLAST. Like we learn that in BLAST, if we want to align this reads to the database, instead of aligning the whole sequence, BLAST is going to look for a certain short subsequence called word, which is a subpart of this DNA. And then can try for the perfect match for this word in the database. And that's because finding a match for a short sequence is more easier. It's easier than aligning the large sequence. And the idea is, if this ribosomal RNA sequence, if it's going to match to some RNA in the database, then a shorter segment, like maybe a 6-mer, like 6 nucleotide from this sequence, also gonna match because it's a shorter sequence. Like if the whole sequence match, the shorter part also have to match. So they are framing the alignment as a different problem. Instead of trying to find the actual alignment, like using dynamic programming, they're going to ask, if I extract all the 6-mer, like all the 6 nucleotide subsequent from this read, can I find a certain RNA in the database that contains all these patterns? If I have the RNA, ribosomal RNA in the database, and it contains TGC-TTA, it contains GC-TTAT, it contains CTTATG. If it contains all these patterns, it's very likely that that RNA is going to match my read because it matches every 6-mer in my reads. So the chance that it's going to probably match the whole sequence. So if we use the probability form that we saw from the last few lectures, what we want is we have the sequencing data. So we see all the Kmers, like W1, W2, these are all the Kmers. We observe this. What we want to find the genus or the taxonomic unit that maximize this. We want to find the genus that most likely comes from our observed read, that correspond to this observed read. And again, we know that this is difficult to calculate. But if we know the genus, like if I give you the genus, you can look up all the ribosomal RNA from that genus. You have the database. You look up for that genus, you got all the ribosomal RNA sequence. If I ask you from that sequence, from those ribosomal RNA, what is the likelihood, what's the probability that your sequence data is going to produce this word? This is easy to calculate because you have the entire database. You have the entire book. If I ask you what's the probability of seeing a certain word in the book, right? If you have the book, then you can tell me directly how many occurrence of this word in the book. And you can give me that probability. So this is easier to copy. So this can just reuse the concept of Bayes rule that we saw last time. Okay, to give you some more visual correspondence. So we want to try to identify sequence in the database that match to our read. But instead of doing the direct dynamic programming, sequence alignment, we're going to generate all the possible shorter fragments from our read. Like these are all shorter. And we want to identify like which species or which genus in the database that contain these short signatures, like short words, short kmer. And we can count. Like, for example, if I have a certain sequencing data, and in my database there is a coccidioides, it might contain 112 of my kmers, but actually I might have like 10,000 kmers. So if it only matches to 112, it's probably not a good match because I have 10,000 kmers. And this genome, this ribosomal RNA, only match to 112 of them. So this is probably not the correct answer. But if I find human polyoma virus 2, it matched to 7,000 of my short fragment. Then this is probably the good one. I have 10,000, but this species matched to 7,000. So the rest might be due to variation or sequencing error. Then based on this result alone, I can have a good guess, a good estimate that my species or my genus is going to be this pathogen. So this is much faster than dynamic programming. So people are using this a lot to get like a good first estimate in our sample. Like if we can classify all the reads very quickly, whether it's human or non-human, you can get a good understanding of how much contamination you are having in your sample, which abundant bacteria are present in your sample, as a rough estimate before doing a full analysis. And also, people are using this as a way so that when we have many reads, like we have millions of reads that are mapped to the same species can be grouped together. If we know that these 7,000 reads maps to human polyoma virus, when we want to do the assembly, like we want to reconstruct the organisms, we don't have to consider human reads with the virus read, with the viral reads. If we want to reconstruct the virus, we just consider all the reads that are mapped to the virus. So this can also help, this technique can also make sequence assembly much easier. Instead of having to assemble a million reads together, you might group the million reads into different bins. One bin for human, one bin for E. coli, one bin for the virus. And then you can do assembly separately for each bin. Okay, so we just learned about taxonomic classification using alignment or using a new technique, like using Kramer patterns to classify the taxonomy. So now we are going to talk about, once we have the sequencing data, like we learned from prior lecture that we have to do some processing. So we also have to do some processing on reads that we get from metagenomics. And it's quite different from what we saw before. So first, if we do amplicon sequencing, like ribosomal RNA sequencing, when we amplify the hypervariable region a lot, like to get all the reads, sometimes during the PCR amplification, you can get a incorrect product. So you can get a chimeric product, where the front might come from a certain ribosomal RNA, but the back come from a different ribosomal RNA. So you get a chimeric or a fusion product. So what people have to do when we analyze 16S RNA sequencing is we want to remove these reads. And to do this, we use the same technique when we talk about the detection of gene fusion or the detection of chromosome translocation. We have a situation where the front, like if you do a parent sequencing, the forward read and the reverse read are going to map to two different things. If it's a gene fusion, the forward and the reverse read might map to different chromosomes. But in this case, the forward and the reverse read is going to map to RNA sequence from different organisms. So we have a discordance between the mapping of the forward read and the mapping of the reverse read. So what people did is very simple. We take the read, like the whole length of the read, and we cut them into pieces. For example, 4 pieces. So we basically just chop the first part, second part, third part, and the last part. And we do sequence alignment to the rRNA database. If all the 4 pieces map to the same species, then we are cool, right? That means the whole thing comes from the same organism. But if different chunks, like different section of your read, map to different species, map to different genus, then that means you are having a chimeric read. The first part map to certain bacteria, the last map to a different bacteria. When we have this result, we can reject, we can remove these reads. So this is the same concept as when we do gap-led alignment or when we detect a gene fusion. The second thing that's very interesting is when we sequence human DNA, for example, like, because people have been studying human for a long time, you don't have to guess like how many reads do I need to study the exome. You can read human exome sequencing papers, and you have all the information you need, like how many million of reads do you need to characterize a certain mutation up to a certain allele frequency. But for metagenomics, if you study water sample, or if you study gut microbiome, or if you study go to the wildlife sample, you have no idea how many bacteria are there in the sample. Right? How much read do I need to study? So this is always a question that people who do metagenomics have to ask. Do I know the complexity of the sample that I am sequencing? And a quick answer is we don't know. But once we sequence, so before we sequence, we have no idea. Right? But once we sequence, there is a way to check. So this graph called a rarefaction curve. The idea is that you already did the sequencing. So let's say you have 20 million reads like here, and in your sample, you can identify certain number of taxa. You have a certain number of OTU. What you can do with your data is you can, instead of considering all the 20 million reads, you can try to consider just 18 million reads, 15 million reads, 10 million reads, 5 million reads, 3 million reads, 1 million reads, 100,000 reads, 50,000 reads. And as you consider less and less of your data, you're going to see less and less of the diversity. You're going to see less and less of the OTU. The cool thing is, if you have enough sequencing data, that means you have plenty of reads to cover all the OTU. If you lose some sequencing data, you're going to see all the OTU. So the graph is going to go down very slowly. If you see the graph being very plateau, like very flat, this means that you have enough sequencing data. If you lose some data, it's okay. You can still recover the same OTU or the same bacteria with different read. You lose some read, you can still see it with a different read. But if you are here, when you lose some read, the number of OTU goes down very quickly. This means that you have a lot of species, a lot of bacteria, that you might have like only 1 or 2 reads that can tell you that they are here. So if you lose those information, you no longer detect them. So if you see a graph, a rarefaction curve, that's a very steep, that means you don't have enough sequencing data. And this is a signal that you should go back to that sample and sequence more. But if your data are very flat, like this, then this means you are good. You are good to go with the bioinformatics analysis. So this is very important because for metagenomic sample, we never know what is the complexity, which species or how many pathogens are we expecting to see. So after you get the sequencing data, doing the rarefaction curve is very important for you to tell. Okay. So rarefaction curve only focus on the number of OTU, like how many species do you see. In the technical term, this is called the richness of the sample. How rich is the sample? It's the number of distinct species or the number of distinct OTU that you can identify. But sometimes when you study ecology, like you want to understand the composition of bacteria in the sample, you also want to look at the evenness as well. So if something is even, then that means everything is of equal proportion. If it's not even, then you might have one species being very high and the other species being very rare. So in terms of the complexity of the sample, so there are many metrics, but to understand some of these ideas, we're going to focus on just the richness, which is the number of species, and the evenness, which is how the species are distributed. Are they evenly distributed, or are they very skewed towards certain bacteria or certain species? So there are 2 formulas here that I include. One is the Shannon entropy or the Shannon diversity index. I believe this one you might have seen in a different context before. And this is the formula. the proportion of each bacteria times the natural log of the proportion of that bacteria times together, and then sum over all the bacteria that you found. So entropy is going to be highest, going to be maximized, if you have an even distribution. If all your bacteria are of the same abundance, like 1 over S, everything being equal, and this is your maximum entropy. So entropy being high means that your bacteria are evenly distributed. But if your sample is colonized by a certain bacteria as a major taxon, and the others are very rare, very low, your entropy is going to be very low as well. So the entropy can give you an idea of how even or how skewed your sample is. Similarly, you might see some people use what's called a Simpson's index. It's just 1 over the proportion square, sum over all the bacteria. So again, this score is going to be maximized because this is on the denominator. Right? This is going to be maximized if the denominator is small. And it's going to be small when everything is equal. If all the species have the same abundance, the Simpson index is going to be the highest, which is just S, the number of taxa. And if you have one dominant bacteria and the rest being very rare, you're going to get a score of 1. So these kind of mathematical formulations were selected to describe the evenness of the sample because they have a good property that if the samples are even, like the distribution is or the abundance are the same, you get high score. If this distribution is very skewed toward a certain bacteria, then you get a low score. So both of them have the same property. So that's why people use these metrics to characterize the complexity of a microbiome sample. Okay. Another tool that you need is how you can compare two metagenomic samples. How can you say that gut microbiome from patient number 1 is similar to gut microbiome from patient number 2? The simplest metric is called the Baker-Curtis dissimilarity. Some of you might have seen this before, and some of you might have seen this concept under different name. So intersection over union. If we have two sets, if we want to say if these two sets are similar or not. If the two sets are similar, they should have a large intersection. So intersection divided by union can tell you whether the two sets are similar. So Baker-Curtis utilize the same idea. If you have different kind of animal or bacterial composition in the two samples, so these are basically two sets of animal. We want to compute the intersection. So the intersection between these two would be 1 chicken, like 2 goats, and 1 mouse. So that means the intersection is 4 animals. And the union, the whole thing, contains 11 animals. So the Baker-Curtis dissimilarity in this case would be 1 minus 2 times the overlap, the intersection, divided by the number of animals in both groups. So in this case, it's going to be 3 over 11. So this is very simple because when you have the taxonomic classification, like let's say you change chicken to E. coli, you have 2 reads mapped to E. coli in sample 1 and 1 read mapped to E. coli in sample 2. You're going to count 1 read from E. coli as the intersection, as the overlap. And the first sample is going to have another read for E. coli being unique to sample 1. So this concept that you can change from animal to bacteria is going to work out the same. Instead of the number of animals, you can think of this as a number of reads. You have different reads mapped to each organism in your data. So Baker-Curtis is very simple. Once you classify all the taxonomy, you just count like how many reads you have mapping to different organisms. But Baker-Curtis is also very limited. Anything that's simple is going to be bad in certain circumstances. So let's say you have a different sequencing depth. In the first sample, you only have 6 animals, like you have 6 leads. In the second sample, you have 18 animals or 18 leads. You have 3 times the sequencing depth. Because Bray-Curtis count the number of leads, right, you can't really normalize the data easily because you need the number of leads. So in this case, the overlap is going to be 2 chickens, 3 goats, and 1 mouse. The overlap is just 6 because sample 1 only contains 6 animals, only 6 leads. Your overlap cannot exceed that. And the intersection is very large. It's 24. So you have a Bray-Curtis dissimilarity of 1/2. Even though, like, if we think about the composition, both of these samples have 2 chicken per 3 goat per 1 mouse. They should be the same. You just sequence one more than the other. But with Bray-Curtis, you are going to see this being very different because you cannot account for the change, the difference in sequencing depth. So this is one limitation of Bray-Curtis dissimilarity. So whenever you use this, or whenever you see anyone using this, always check if they can have this problem of sequencing depth being higher and lower across samples. The second limitation is the similarity that considered overlap by Bray-Curtis dissimilarity doesn't consider taxonomic information at all. So I'm going to illustrate this by making some of the code black and white. So in this case, you're going to say that, okay, I have between sample 1 and 2, 1 chicken and 1 mouse, because you have white goat and black goat being different organisms. But we know that from, let's say, from genetics information or from taxonomic information, sample 1 and 2 should be more similar than 1 and 3, because a goat being black, the genetic difference should be small. But from goat to a pigeon, like the genetic difference should be very large. So Bray-Curtis also doesn't take this into account. Even if you have a closely related bacteria, as long as in your data they are called different genus, like different species, Bray-Curtis is going to treat them as completely different. So that's a limitation. So what can you do better? There is another matrix called UniFrac distance. And this is based on phylogeny. So first, you're going to have to do some more work. For Bray-Curtis, you just assign the taxonomy and then you count. For UniFrac distance, you're going to have to take your sequencing data, maybe assemble them into genes, into genome, and you have to do phylogenetics. You have to do some evolutionary analysis. You reconstruct this tree. And from this reconstruction, what you can calculate is the fraction of the branches where downstream from that branch, if you see leads from both samples, then you count this branch as being a chair between the two samples. So to compare, like between A and B, in A, like your sample 1 and 2 contain many similar species, many similar organisms. Your phylogenetic tree have a mix of sample 1 and sample 2 kind of throughout the tree. So this means if you see organism in sample 1, you also see a relative of it in sample 2. But if your sample are completely different, let's say different bacteria entirely, bacteria in sample 1 are similar to each other, and bacteria in sample 2 are similar to each other, but they are not similar at all. In this case, the UniFrac distance is going to be 0. In the example on the right, for the B example, none of the branch is shared between sample 1 and 2. Each branch either only contains sample 1 or sample 2. But on the example on the left, in example A, almost every branch, if you look further down the line, it's going to contain both organisms from sample 1 and 2. So UniFrac distance takes into account the phylogenetic or the genetic similarity when they quantify the similarity between samples. So this is much better than Bray-Curtis. But you have the burden of having to reconstruct the phylogenetic tree. So in this sample, it only contains like 20 organisms. But in your data, like if you study a microbiome, you might have hundreds of organisms. That means you have to reconstruct very large phylogenetic tree. But from last lecture, like we know there are tools like FastTree where you can reconstruct this tree very quickly. And UniFrac, this sense, in this example, doesn't really take into account the abundance. We just reconstruct the phylogenetic, the presence or absence of the species or the organism. But in your data, you might have a lot of weeds. Like this species might be a dominant bacteria in sample 1. But this species might be a dominant bacteria for sample 2. In that case, even though you have a shared phylogenetics, but the abundance doesn't correlate. In sample 1, you have high abundance of this bacteria. In sample 2, you have high abundance of this bacteria. So UniFrac, this can be modified. So that each of these branches is going to take into account the abundance of the organisms when you calculate the similarity. So if you have abundant bacteria also being the same, that's even more similar than having the same bacteria but different abundance. Okay. So with amplicon sequencing, especially 16S ribosomal RNA data, these are the things that you can do. You can classify the taxonomy, you can calculate the similarity between two compositions, between two samples. So this is the easy part of metagenomics. So now let's see the difficult part. So the shortcut metagenomics, where we sequence all the genes in the sample. So we have reads coming from different genes, different organisms in the data. So there are two possible ways to go about this. If you have long read, you might be able to do assembly directly. We have long read data. We can reconstruct the assembly very easily. But if you have short read, we know that from our assembly lecture, even doing assembly for a single genome is difficult enough. If we have to do assembly where different reads might come from different organisms, if you connect reads from the blue organism to reads from the red organism together, you get incorrect assembly. So you have even more ways to go wrong. So in this case, the first step in almost every pipeline is called binning. So binning is the idea that if we have somehow an algorithm that can group reads from the blue genome and from the blue organism, if we can somehow group them together, if we can group all the reads from the red organisms somehow, if we can group them together, then we can analyze them separately. And that can make the problem much easier. So basically we transform metagenomics situation into a single genome situation. We just need to do assembly multiple times. But each time is a single reference, single genome. So what can you do? There are two ways, at least two ways, that we can bin the reads so that hopefully reads coming from the blue organisms will be grouped together, reads from the yellow organism will be grouped together. The first one utilize the same idea of K-mer that we see before. We saw the idea that if we have K-mer from our read mapped to a certain taxonomy like using ribosomal RNA database, we probably can classify that read as being from that taxonomy. In this case, because we don't know the taxonomy yet, for the case of ribosomal RNA, we have the database. But remember that for shotgun metagenomics, this read can come from any gene, even from non-coding region. So it can be anything. We cannot classify the taxonomy like before. But what we can do is, because different bacteria, because different genome, can have different GC content. So basically, different genome have different sequence. So if you characterize like the K-mer frequency, how many AATA do they have, or how many GC content do they have, we know that different species, different organisms have different value of these signatures. So what people did is, let's say we calculate within each read, we calculate the frequency of K-mer, FOMer, AAAA, AAT, AAC. We have the frequency. And our assumption is that if we have reads coming from the same blue organisms, they should have similar profile, either similar GC content or similar former profile. So what we can do is, if we see multiple reads like with the same GC content, with similar former profiles, we might want to group them together because they might come from the same organism. But we know that something like GC content is not all fully specific. Right? You can have different type of genome entirely with the same GC content. So the second evidence that people use to bind the reads together is the abundance of the read. So let's say you have a certain read, a certain sequence. Let's focus on the sequence. For each sequence, you can have multiple reads because you have multiple molecules of the DNA and RNA. So if you see several sequences where your read count for those sequences across sample, it might take some imagination. Let's say we have 4 samples. Sequences or reads that originated from the blue organism is going to be low, high, high, low. Because in different samples, you have different proportion of the blue organisms. So any reads that are generated from the blue organisms, you're going to see a lot of them in sample 2 and 3, a medium amount in sample 1, and low amount in sample 4. So if you consider many sequences, and you look at the read count that map to the sequence, if you see the same pattern, medium, high, high, low, it's likely that these are from the same species because the abundance can attract over sample. Let's take another example. Let's say we look at reads that might come from the black genome, the black organism. So this read, this sequence, should be high in sample 1 and 4 and low in sample 2 and 3. So we can use this pattern to group reads, to group sequence together. And hopefully with the combination of the GC content or the KMER profile together with the abundance that tracks across sample, these two together should be able to bring you the read from red organism together, the read from the blue organism together, the read from the black organism together. So this is kind of what people did to make metagenomics analysis simpler. And after this, you can apply what you know about single genome situation. You can do assembly regularly. You can do alignment, you can do everything afterward. So just to give you some kind of mapping of the steps and the tools in metagenomics, because there are a wide variety of them. So in these next 3 slides, I'm showing you the 2 pipelines. Again, one is the amplicon sequencing pipeline. So you pick a gene, you amplify it, and then you try to analyze the taxonomy composition of the sample. So you pick your marker gene, you sequence it, PCI amplified sequencing. You can do QC like before, because these are sequencing data. For processing, again, you can do trigonometric, like to cut out the adapter, same as before. But now you want to classify the taxonomy. There are 2 steps. First, we want to cluster similar reads into OTU first. So read that looks very similar, like maybe 99% similar. We want to group them together. And this is to reduce the amount of analysis that you need to do. Let's say you want to do taxonomic classification by alignment to the database. You generate 10 million reads. If you align all of them, like you are going to do 10 million alignment. But if you know that some reads are very similar, like 99% similar, they are going to map to the same taxonomy anyway. So the step of OTU estimation here is where you reduce the redundancy. If you have sequencing reads that are almost identical, you're going to collapse them. Instead of aligning all of them, you're going to collapse them. And then you might have, let's say, instead of 10 million reads, you might have 500,000 that you need to do the alignment. So this will reduce the computational time. But once you arrive at the taxonomic classification, so we use the 2 techniques that we mentioned. You can do sequence alignment to the database, or you can look at the KMER profile. to classify the taxonomy directly. And the last step here is something that, from what I heard from people in the field, it's not very accepted, but it's something that you can try to do. So one limitation of analyzing amplicon sequencing data is you know the ribosomal RNA, so you know the taxonomy of your bacteria. But you don't know anything about the other genes that bacteria may have. You don't know the plasmid, you don't know the rest of the chromosome. But sometimes people want to be able to interpret if a change in taxonomy in your patient and healthy sample, healthy people, you might see a change in certain bacteria. They became more abundant in the patient. Sometimes, as a biologist, you want to be able to interpret. So, what kind of functional impact does the change in this bacteria have on the disease? But because you only know the name of the bacteria, you cannot tell anything about its function. Like you don't know which metabolite that it can process. You don't know which toxin that it can produce. So what people did as a last possibility here, the functional prediction. So what people did is they go through the database again, database of complete genomes, not the database of ribosomal RNA, but the database of complete genomes. And people ask, let's say I saw an enrichment in Shigella in the patient, and I want to know, like, does Shigella encode certain toxin, or does Shigella encode certain enzymes? Even if I don't know the actual genomic sequence for the Shigella strain that I discovered, I can still go to the database of the genomes for other Shigella that other people have sequenced and ask, do Shigella's genome that other people have sequenced, are they enriched with certain toxins? Are they enriched with certain enzymes? And I can predict that, oh, maybe in my patients, the Shigella genus that occur there, they might have the same toxin, they might have the same enzyme. So this is like a large effort to try to understand the function of the microbiome that we just discovered without actually knowing any gene. Like, without knowing any gene, we just guess that if other people have seen these genes in the genus, then maybe my patient also have them. So it's not widely accepted because this is a big assumption. You are assuming that the cause of the disease is just the presence of that genus with nothing special. But maybe in your disease, that genus Shigella alone might not be enough. It might have to acquire certain plasmid or acquire certain gene in order to cause the disease. And we don't know because with amplicon sequencing data, we only know one gene, the ribosomal RNA. Okay. So these are the key steps that we mentioned in today. We talked about the processing, the removal of chimeric sequence, and the alignment of chamber-based classification for the taxonomy. For shotgun metagenomics, where we have sequence, where we have reads from all the genes, I mentioned that there are two ways to go. We can assembly, or we can, if we have a long read, or we don't need to do assembly, then we can also try to characterize the taxonomy. The key idea here that I want to emphasize is this step. The blue box here, this is binning. So before we apply any complex bioinformatics analysis, what we want to do to metagenomics data is to bin them to group reads and sequence that likely come from the same organisms together so that they become easier to process. And in this diagram, you don't have to worry about too much details, but if you ever need to analyze the data, all these grays, these text in gray, these are the bioinformatics tools for each step. So you know what to do next, and you know kind of which tools to use. But just to show you the flow. Okay. And another keyword that I want to introduce. So once you do the binning, you get all the reads that likely come from the same organisms together. Then you want to do the assembly. You want to assemble the genome. In this case, because the assembly is from metagenomics, which require a lot of processing beforehand. The binding part might not be perfect. So people will call this assembly MAG, Metagenomics Assembled Genome, to designate the fact that these are not pure isolate. So these assembly might contain errors in them. So anytime that you see MAG, these are genomes that are not from pure isolate. Pure isolate means you have one species and you sequence that. That's a pure sample. You can reconstruct the genome without having to worry too much about contamination or DNA from different species. But when you see an assembly or a genome that's based on metagenomics or MAG, you have to be careful that the data, that the genome assembly that you have might contain errors, might contain sequence from a different species being incorrectly assembled. Okay. So the last thing that I want to introduce for today is when we study microbiome. So actually when you study anything, what we always worry about is the batch effect. If we analyze the same sample twice, we will get the same result. It's an issue in every wet lab experiment, but it's an especially important issue in metagenomics or microbiome study. So in this example, they analyze the same sample. So let's say the same gut microbiome sample, the same stool sample, with different DNA extraction protocol or different sequencer. And what you can get is very different result of the bacterial composition, because different extraction protocol and different sequencing platform have different bias. They will enrich certain bacteria than the other. So this is a caution if you want to do like a meta-analysis by comparing sequencing data from one paper to another paper. You have to be careful. For metagenomics, any small differences can make big change in the result. So the best would be, if you have to design your metagenomic analysis, you have to include all the control, everything in your design, and you should produce the data yourself, and not. So we will see later on that for other omics like transcriptomics or proteomics, it's sometimes safe to download data from different papers and analyze them together. But for metagenomics, it's not. So this is something that I want to emphasize. And I have direct experience with the data that are produced at our universities. So the color here designate data from different batches. So there's one batch here where you can see from the heat map, the y-axis is the different organisms, different OTU. For most of the batches, it's not a problem. You see the same abundant bacteria. But somehow, for the red batch, for this one, to this day, we still not sure what happened. But you can see that the dominant bacteria completely changed to something else, even though these are the same patient cohort. So that's why we have to be very careful when we design a metagenomic experiment. So, yeah. Okay, so that's everything that I have for today. So see you next time.